good, dear friend of mine and somebody that I've been working with and admiring their science for a very, very, very long time. He's currently at the University of Virginia. And his talk title is Systems Genetics Analysis of Smooth Muscle Cell Phenotypes in Diverse Ancestries, which you can tell by the title. Obviously, he works in genetics and genomics and the functionalization of thereof. So someone who's really taught us a lot about why all these SNPs and variants in the genome actually matter, particularly with a focus on smooth muscle cells. And his work has, been just so in depth and a real tour de force when you're working with humans and you're working with human samples. You can't just have, as we say in my lab, another cohort that we just kind of keep doing over and over. It becomes very, meticulously planned and very, very wonderfully executed experiments, which you'll see. So he's an associate professor in biomedical engineering at the University of Virginia at UVA, and in the Center for Public Health Genomics. And he's the chair of the Diversity, Equity, and Inclusion Committee in the Biomedical Engineering Department. He received his PhD from the University of Pennsylvania in bioengineering and did human genetics at UCLA together with some amazing geneticists there, Jake Lucis and others. So his lab, as I said, focuses on understanding the genetic mechanisms of cardiovascular and metabolic diseases, so both. And not surprisingly, his work is very, very well supported by the American Heart Association, the American Diabetes Association, Duke, the NIH. There's just it goes on and on and on and on. A fellow of the American Heart Association. He's a chair of the program committee of the American Society of Human Genetics and part of the leadership team of the HHMI supported Driving Change program, which is very important and I think just as transformative of the work that Mete does in his lab is the work that he does to make sure have inclusion and really promote students of all backgrounds and those who are generally underrepresented and excluded from science. And Mete is a fabulous mentor and a real supporter of all of those in science at the University of Virginia and others. So it is my distinct pleasure. Welcome, Mete, and we look forward to your talk.
Wow. Thank you. That is a very generous introduction, by the way. I really, really appreciate it. And I also really appreciate the very generous invite. It's really fun to be here. If you are a human geneticist, I know she's not here. And if you're, doing cardiovascular genetics, first of all, you are a fan of Ruth McPherson, who happens to be here, obviously. So Otto Heart Institute is a place that I've known for, 15 years. It's really, really wonderful to be able to visit finally. So let me get started. But what I want to say is I have different chapters and I'm happy to skip, say, one in case you have questions and want to interrupt. I think it's much better to do it that way. I don't have to go through everything. So my lab studies the genetics of coronary artery disease, and I don't need to preach to this crowd that it's the leading cause of death in the world, and the underlying pathology is, this process that we call atherosclerosis. So I'm going to use the laser pointer just so that the people online can also see. I think so that they can follow the talk. And as you also know, that coronary artery disease has both environmental risk factors like our lifestyle and smoking and physical activity, but also a very strong genetic component. The heritability studies that are done in populations, in twin studies and families point to 40 to 70% heritable component to the risk for having a heart attack or stroke and coronary artery disease. So as I mentioned, the genetics of coronary artery disease really got started here in, I think, 2007 with the discovery of the 9p21 locus. And of course, it's taken off from there. This is the most recent Manhattan plot of the coronary artery disease GWAS. So this was a study that was done in more than 1 million people. And you see these concentric circles. Each of these peaks are the regions of the genome that are associated with coronary artery disease. This is starting with chromosome 1, going all the way to chromosome 22. And the inner circles are actually studies that were done in people with different ancestries, like African ancestry or Asian ancestry, etc. And there are now, including all these large, different ancestries and big studies, about 300 genomic regions that are associated with increased risk for coronary artery disease. And if you look at those regions a bit more closely, you can see that some of them are also associated with very well-known risk factors like lipid metabolism, like blood pressure, like adiposity, etc. So we can say that those loci are affecting these risk factors, and that's the mechanism by which they affect the risk for coronary artery disease. But if you look at other loci, we have so many unknowns. And even when we, point to things like proliferation and transcriptional regulation, they are in a way a bit meaningless because you don't know what transcripts, what are they regulating in which cell types, et cetera, et cetera. So basically, the way I view GWAS is they are very solid results that are done in millions of people. They generate hundreds of hypotheses about the disease process. So one of the interesting aspects that we have known for many years of these GWAS loci is the fact that very, very few of them are in actually coding regions of the genome. So only 1% of all the single nucleotide polymorphisms that are associated with coronary artery disease are in exonic regions that lead to a change in the amino acid that potentially changes the protein function. 99% of them are in regulatory regions of the genome. So what are they doing in these regulatory regions? So there are two potential mechanisms of what they could be doing. One is regulating expression. So imagine a case where there is a single nucleotide polymorphism. There are people with an A allele and there are people with a C allele. And this happens to be in the promoter region of a gene that affects the binding of a transcription factor. Very simplistic molecular mechanism, right? And if the C allele leads to perfect binding of the transcription factor, well, that will lead to higher expression of genes, right? And if the A allele disrupts transcription factor binding, that will lead to low expression of the gene. So this is one molecular mechanism by which these non-coding regulatory SNPs might be affecting the disease process. Another one is by affecting the splicing of the gene. So imagine again, these SNPs sitting in splice acceptor, splice donor sites, they can lead to the abundance of different transcript isoforms, right? So these are two potential mechanisms by which these loci might be affecting the disease process. So you can study this, one by one, or you can study this in essentially across the genome, which is basically the approach that we take, and we call this systems genetics. So you have this DNA variants at the genome level, and they are somehow associated with the disease, right? But you have obviously all these intermediate molecular phenotypes that kind of carry this information that's encoded in your genome to affect the cellular processes that then affect the disease, right? So this is the approach that we're taking. And when I started my lab, basically we said, okay, we know that atherosclerosis is obviously a vascular disorder. A lot of people have studied the loci that are affecting the lipid metabolism. So that's fine, but a subset of these loci should be affecting the vascular wall processes. And this is a cross-section, a cartoon version, obviously, of an atherosclerotic lesion. We know that smooth muscle cells play an important role, and some of the fantastic work here also shows that, not just obviously the genetics. They're in this quiescent state in a healthy artery that then change a phenotype, become more proliferative, calcified, etc. They migrate from the media to the intima, etc. So we thought, okay, some of the risk variants are potentially affecting this process, and that can help us solve some of the mystery of the unknown loci. How do you test a hypothesis like this? So we have been collecting basically aortas from heart transplant donors. So these are people who unfortunately died in a motorcycle accident or a traffic accident, but they're otherwise healthy. They're healthy such that their hearts can be transplanted into individuals who have, let's say, heart failure, right? And as you can also imagine, it is impossible to biopsy, right? Coronary arteries or aortas from healthy individuals, but you inherit your genetic variants from your parents. So if you have a risk allele, it is working somehow since your birth. So it is actually important to study the function of these risk variants in different stages of the disease. So normally a surgeon will cut pieces of this aorta to fit the heart into the recipient, and they would discard it. So we took smooth muscle cells. We also took endothelial cells, but we haven't worked with them, and we cultured them. And these are from 150 donors, and majority of donors are male. This is in line with the heart transplant donor pool, at least in the United States, where the majority donors are from men. So we genotyped these individuals, whole genome genotyping, for about 6.3 million SNPs. And one of the first things that we did was to actually cluster the genotypes of these individuals with reference populations. So there's a project called 1,000 Genomes Project that went out and collected genomic information from people around the world, and there are 20-some different populations that are represented. And these black dots are our donors, and these are basically the continental populations. So what you can see is that about half of our donors are of European ancestry, but actually majority are of non-European ancestry. And this is actually also something unique because most of the GWAS is done in people of European ancestry. So these actually individuals help us to increase the genetic diversity of the genetic SNPs that we're studying in our population. Okay. So we have these donors, we have these cells. What do we do with them? So we performed essentially what we call GWAS in a dish. Okay, so we took these cells and we measured 3 different cellular phenotypes that we thought are relevant to atherosclerosis. So migration, proliferation, and calcification. Of course, there are many more, right? Some people would have wanted us to study apoptosis. Some people would have wanted us to do X, Y, Z. But we had to set up basically high throughput assays and do it in a frankly reasonable way to be able to characterize all these cells. And the other thing we did was to study these under different conditions. So for example, for migration, we know PDGF-BB, the cytokine PDGF-BB plays an important role. So we studied migration under just baseline control conditions and in the presence of PDGF. We studied proliferation in response to 3 separate cytokines because we know they simulate different pathways and affect different parts, if you will, of the cellular signaling that leads to proliferation. And we studied calcification in 2 different conditions. So these are the cellular phenotypes that we studied. And then we also studied molecular phenotypes. If you remember these layers that I mentioned, we also want to understand the molecular mechanisms of how these loci are acting, right? So we looked at chromatin accessibility, we looked at gene expression, and we looked at secreted protein abundance. And we did this in 2 different conditions. Now, this is a bit simplistic, of course, but we culture the cells either in the presence or absence of FBS, which are, kind of inducing a quiescent and a proliferative phenotypes. Now, I want to caution, I'm not saying this is capturing what is exactly happening in the vessel wall, right? But clearly these are different, aspects of the cellular phenotypes, and it's been studied in the literature very well, basically using these culture methods. So I'm going to mostly focus on the gene expression part today, but happy to answer, of course, other questions too. So let's first talk about proliferation. I'm not going to talk about every single phenotype, but I want to give you an understanding of how we perform these experiments. So for example, for proliferation, we take these, there's the high throughput 96-well assays that you can do. So we culture one donor in 4 different conditions, and you can see that each condition has 6 different technical replicates. And then because we cannot do everything at the same time, right, we have to use basically a reference donor, which we use in every single experiment to normalize it, to get rid of the technical aspects, technical variation. And when we do that, we can, for example, calculate proliferation either with respect to the reference donor, or we can look at the response to these cytokines with respect to the control conditions for a given donor, right? So again, like I won't tell you every single phenotypic measurement that we did, but overall, we have 7 phenotypic measurements in proliferation space. We have 3 different migration phenotypes and 2 different calcification phenotypes. So when we did this, right, of course, we're doing this frankly blindly, right? We're hoping we're gonna see some associations. And the first thing to do was to look at the phenotype distribution. This is what the proliferation phenotype looked like. So you can see we ranked the donors. They are on the x-axis. All of these are the 150 different donors. And you can see that there's a huge variation in the amount of proliferation that these donors have under just basal conditions. And this variation holds up for basically every phenotype that we study. There's a huge variation among the donors. So the first thing we asked was, okay, is this age related? Is this sex related? Is this ancestry related? And no, it does not explain, none of these variables explain this phenotypic variation. So we thought, okay, then maybe there is really the genetic variation of these donors that's leading to this much phenotypic variation across the donor pool. But I wanted to be sure that this was indeed the case. So we did one study where we took 4 donors. We cultured them and froze them in 2 different passages. And we did these experiments in the 2 different passages, in the 2 different donors on 3 separate occasions. So this took basically like one undergrad, a whole semester to do all of these things, right? So we're deliberately introducing technical variation to the experiments, right? Okay, so we measure the 12 phenotypes again, and we then do hierarchical clustering to see what it looks like. So the hierarchical clustering showed that the phenotypes basically cluster based on the donors, right? So this again showed to us that indeed the phenotypic variation is due to the donor-to-donor variation, and it's not the technical variation that's causing this. Okay, so I'm going to skip a lot of things, you know, because part of this has been published. But of course, we basically did a GWAS on these donors and the phenotypes and found things that are already well known in the literature, which is kind of our positive control. But the, you know, interesting, I guess, thing for us was, do the coronary artery disease loci actually associate with these phenotypes, right? Okay, so I'm going to summarize that in this kind of complicated and ugly heat map, but bear with me, I'll try to explain it, okay? So first of all, these are the phenotypes that we measured, the 12 phenotypes that I keep mentioning about. And these are at the bottom are the loci that are associated with at least one of these phenotypes. Okay, there were 79 of such loci. And first of all, very few of them were also associated with lipids. I think just 1 or 2 of them. So meaning that we are actually truly capturing the loci that are hopefully affecting the vascular wall biology and not other phenotypes, right? Second is you're going to notice that some of the loci are associated with just one trait, right? In this case, with calcification and another measure of calcification. But there are also some loci that are associated with multiple phenotypes, like proliferation and migration, right? So it's pretty complicated in terms of how some of these loci might be affecting the smooth muscle cell biology. The second thing is we colored these based on the direction of effect with respect to the risk allele. So if you have blue, and if you're a person carrying the risk allele, it means that risk allele leads to less of that phenotype. And if it's red, it means it leads to more of that phenotype, right? So again, what you can see is that some loci, for example, increase proliferation, some loci decrease proliferation. And this really goes on to, just to show the complexity of the genetic effect on the smooth muscle cell function that might be relevant to atherosclerosis. So I showed you, of course, the cellular phenotypes, but how about the molecular phenotypes, right? Because that's also something that we're interested in. Obviously, we want to understand the molecular mechanism. So remember, we did all this RNA sequencing. And the way we did it was we cultured the cells in 2 different conditions. So now we have 302 different cell cultures. And we did ribosomal RNA depletion because we actually wanted to capture the long non-coding RNAs as well. And if you use, poly A, you don't always capture the long non-coding RNAs. And we did really deep sequencing because long non-coding RNAs are usually basically, lowly expressed in general, right? So to be able to do this, I overspent one of my AHA grants, and I got in so much trouble for that, basically. Okay, so we ended up measuring the expression of about 18,000 genes, right? Most of them are protein coding, but we have about 3,000 long non-coding RNAs as well. So the analysis that we're doing with this is this EQTL discovery. So an EQTL is basically a locus, a genetic variant that affects the expression level of a gene. So we do this for 18,000 genes and 6.3 million SNPs. So we do this, billion linear regressions basically to really find if a SNP affects the gene expression independent of these confounders, right? Confounders basically. And if it is, it should look something like this, right? Where you have an A allele, for example, that leads to low expression compared to the C allele. So let me first show you the general findings. We have, of the 18,000 genes, about 6,500 of them that have an EQTL, a cis EQTL, which means there's a genetic variant that is associated with the abundance of the transcript from that gene. And one of the interesting things that you can see here is that there are phenotype-specific EQTLs, right? So a minority of these are actually overlapping between the 2 conditions. Majority of them are in the unique condition. So this is actually showing the impact of the cellular environment on genetics. We actually formally tested this. And let me show you 3 examples of that. So again, this is a bit of a complicated plot, but let me first focus on this to your, I guess, left side. Is it left? Yeah, your left. So look at this, for example, gene, this NDUFA12 gene. There is an association with this SNP only in the proliferative condition. There's no association at all in the quiescent condition, right? Somehow the genetic effect is only observed in one condition, but not the other. For this gene, ZNF573, the effect is the opposite, right? You have a SNP that is leading to high expression in the quiescent condition, the T allele, that leads to low expression in the proliferative condition. And in this case, actually, these are a bit harder to observe, but you see an effect size difference. For example, in this case, in the quiescent case, the G allele leads to a much higher impact compared to the A allele in this condition versus the proliferative condition, right? So these are obviously quite interesting cases which we consider to be gene by environment interactions in human genetics. Basically, the genetic effect is different in different environmental conditions. So remember, at the beginning, I mentioned that one of the ways that EQTLs might happen is if they affect the binding of transcription factors. And I also told you that we actually looked at accessible chromatin regions in our cells. So we basically looked at these SNPs that are affecting, that are in accessible chromatin regions that might potentially be affecting transcription factor binding. Because we thought that is one way for us to understand this phenotype specific, if you will, genetic regulation, right? And when we do that, obviously separately for the quiescent cells and the proliferative cells, and look at the enrichment of the transcription factor binding in accessible chromatin regions, what we find are these 5 transcription factors that have very significant enrichments in the 2 conditions. For example, this SP2 has actually never been studied in smooth muscle cells. But you can see that there is a very significant enrichment in the proliferative condition compared to the quiescent condition. They're both enriched, but there's a difference in the enrichments. And now actually we're working on this transcription factor. We're doing ChIP-seq and trying to find out where it's binding, etc. But we think that this is one way for us to figure out if there are transcription factors that are actually affecting this the smooth muscle cell phenotypic transitions. Okay, so this is the general I guess picture of EQTLs. The $1,000,000 question, if you will, is do the coronary artery disease associated loci affect smooth muscle cell gene expression? So remember, I mentioned there are about 300 CAD loci, and there are thousands of EQTLs, loci that affect gene expression. So one has to do a pretty strict statistical test to basically to be able to answer this question that I have up there. And the way to do that is an analysis called colocalization. So let me briefly explain this. Basically, this is say a locus on chromosome 7. This is the association with coronary artery disease. These are the individual SNPs in this locus. And you can see the strength of the association in -log10 p-value. And basically, this group that is all red are what we call SNPs in high linkage disequilibrium. So they are inherited together, and it's really hard to differentiate one from the other, which one is causal, which one is not. They're all associated with the disease. And this is the same locus that is associated with the expression of a nearby gene, right? So you have to put these things together, and they have to line across a diagonal for you to say, yes, the same genetic signal is associated with both the disease and also the gene expression. And then you can say, oh, then I predict that this gene might be causal. You're still not claiming causality, but you're narrowing down your search space, if you will. So we did this across the genome using all the coronary artery disease loci. And one thing we found was there were 85 smooth muscle cell genes that are associated with coronary artery disease loci. Now, these genes are, again, we did this based on their location. So you go from chromosome 1 to chromosome 22, and these are all the loci and the genes that are associated with them. And the other thing that we did, similar to the phenotype studies, is we looked at the effect of the risk allele, whether it leads to increased or decreased expression of a gene. So those ones at the top, I don't know, for example, this IRAP2 expression is increased with the presence of the risk allele. And this one, RIC8A, is decreased with the presence of the risk allele. So if you were, right, coming up with a hypothesis on the impact of these genes on atherosclerosis, you can figure that out using this directionality. You can say, if I knock out this gene, then I expect this to happen. Okay, so here's another ugly way of showing you this data. The reason I'm showing you this is because these are the same 85 genes. You can see that you observe the colocalization only in certain phenotypes for some of the loci, right? So at least half of the genes are, active in, both conditions, but they're definitely phenotype potentially smooth muscle cell location or atherosclerosis stage specific effects of these genes on atherosclerosis. So when you have 85 genes and when you have limited amount of funding, you have to prioritize to study them somehow, right? So we collaborated with Minna Kaikkonenmaata from University of Eastern Finland, and she had access to coronary artery single cell RNA-seq data sets. So this is just the overall view of that data. You can see the smooth muscle cell clusters are here, and there's I think this is the endothelial, I can't see it actually really well. Clusters are here. So we basically looked at these 85 genes in this data set. And about 50 of them, it's basically this cluster, had higher expression in one of the smooth muscle cell subclusters. When you do single cell RNA-seq data, you see so many different clusters of smooth muscle cells. So they kind of, okay, we reduced the number from basically 85 to 50, okay. So then my postdoc went through the list and he said, okay, I'm going to work on this thing that there's no publication about. I don't know why he did that, but that's what he did. Okay. So because when there's no publication, that means there's literally no tools to study it, right? Okay, so that turns out to be a long non-coding RNA called SNHG18. He was excited because like, hey, here's a long non-coding RNA. And you can see the expression of this SNHG18 is really pretty specific in the smooth muscle cell clusters of the coronary arteries, right? And if you look at our EQTL data, oops, this froze. That's fun. I'll be patient. Ah, okay. I think I have a lot of images, that's why. Okay, so when we looked at our data, right, you can see that it's actually one of those that act in both quiescent and proliferative conditions, and the risk allele in both cases leads to lower expression of this long non-coding RNA. It's small nuclear, I forgot what HG actually stands for, 18. Human gene, maybe, yeah. Makes sense. It probably is. Yeah. Host gene. Yeah. Oh, I guess I'm gonna slow down a little bit. Remember, I also told you that these loci can be complicated, right? And there can be multiple different SNPs that are in linkage disequilibrium in these SNPs, so in these loci. So we looked at the locus that contains this signal, and I'll show you in this next slide. So is this a snore? It's a snore. Yeah. And, so really, how much is the published one? Nothing. I mean, when we started this, there was nothing on it. I think there was just one study that basically looked at like did the microarray cancers and showed upregulation, downregulation. I mean, and those usually really mean nothing, as you know, right? So this is the signal. You can see that there's quite a few SNPs that are in high LD. Are you coming to? I think it's the laser pointer that's giving you trouble. Oh, I can. Want to just turn that off. I can turn it. And do you see this? If I. No. Oh, okay. But I think it's not working right now, anyways. So I think that's what's giving you trouble. So okay, well then I'll have to explain it. That's fine. Thank you so much. Yeah. So anyhow, so we looked at both the EQTL and the coronary artery disease GWAS signal, and as you can see, they align. And then we looked at the SNPs that are in the open accessible chromatin region. There were many SNPs, but there are 6 of them that are in accessible chromatin regions. So let me turn off the laser pointer as she instructed me. It's gonna come back, but yeah. As long as you don't mind looking at me. So basically, we wanted to identify what might be the causal SNP in this, complicated region, right? And we do what we call massively parallel reporter assays to do this. Are you coming to take a look? Sure. Should I use? Oh,
automatically.
Oh, wow, we lost everyone. Uh-oh. Heard about massively parallel reporter assays. Okay. Anyone else? Okay. So basically, I will just, show you the picture too, but let me explain. Basically, when you want to look at the effect of an allele, right, you clone one nucleotide sequence and another nucleotide sequence, and then, you have a reporter and you try to look at the impact, right, on the allele on gene expression. So imagine doing this for like thousands and thousands of basically SNPs, right? So you're doing like really millions of clonings, if you will, right? So basically we have a 200 nucleotide long, what we call a regulatory element. And right in the middle, you either have an A or a G, depending on the two alleles that you have, right? And that is connected to a reporter. The reporter is actually, what's happening? Should I? Oh, wow, it worked. So the reporter is connected to a barcode, okay? And for each SNP, you create 50 different barcodes. So you're testing each SNP essentially 50 different times. And you put these in a lentiviral vector and you infect your cells. And of course, the cells will go and insert this, regulatory element somewhere in the genome, right? And sometimes there will be one insertion, sometimes two, sometimes three. So what you do is you isolate the DNA and you isolate the RNA, and you first count the barcode that's coming from the DNA to figure out how many insertions happened. And then you count the exact same barcode that is coming from the cDNA, right? And then you say, oh, I have one insertion and 10, of the barcodes. And for the other allele, I have one insertion, but 30 of the barcode. So there's definitely an allelic effect, right, when you do that. And for many of them, there's no allelic effect. But this is one way to find the impact of the SNPs in a massive scale, as opposed to doing it one by one. Because for 85 genes, there are thousands of SNPs that you need to look at. Okay, so let me just show you the 6 SNPs that we looked at in this region, and one of them clearly showed an allelic effect, right? Like really almost night and day kind of, yeah?
Were these 6 SNPs in linkage disequilibrium?
They were in linkage disequilibrium,
so
yeah.
This was to find the causative SNP.
Well, this is the most likely causal SNP, right? I don't ever say causative unless we really test it, right? And the way to test causality is to edit the SNP from one allele to the other and look at the gene effect, right? Basically, which is a really difficult thing to do. But we're doing it, but we're not there yet. But once we found that, we wanted to still provide an orthogonal evidence. So we used CRISPR Cas9, and basically you have a Cas9 that is conjugated to this CRAB domain, which is an inhibitory domain, and you use the guide RNA to basically bring this complex on top of the SNP, right? And what happens is this basically sterically hinders transcription. So when you have a result from this assay, you're just showing that, okay, like there is a regulatory element there. That's all you're showing. But at least the regulatory element is where the SNP is. And again, the way we do it is we have the control cells and we add the Cas9 and the guide RNA separately. Actually, somewhat interestingly, when you just add the guide RNA with no Cas9, there's still a tiny bit of an decrease in gene expression. But when you add both of them, when this machinery is on top of that element, there is obviously an effect. So we clearly have a SNP that is sitting in a regulatory region, right, in the smooth muscle cells. Okay, so what does this gene do, right? That's another interesting thing. Remember, we had phenotype data. So we just simply looked at the correlation of this gene expression with the phenotypes from the exact same cells. And we found that this is correlated with PDGF-induced proliferation, and there's a negative correlation. So we basically took smooth muscle cells, we decreased the expression of this gene by about 60% with shRNA, stimulated them with PDGF, and you see an increase in proliferation, right, in the exact same direction that we would expect from our population study, right? Okay, so as I said, this is coded as a small nucleolar RNA, but of course it's never been studied. So we at least wanted to see where it is in the cell. So we did RNA scope, and this is ACTA2 staining, and this is the SNHG18 staining. And when we overlap them, it appears that, you know, the cells, the RNA is actually both in the nucleus and in the cytoplasm. So this didn't really tell us much. But interestingly, it is outside of the nucleus as well. So that's maybe the interesting finding from here. Okay, so in the next part, we, of course, this is my lab really studies, you know, sex differences in atherosclerosis. So one of the things that we wanted to look at was, are there sex-biased EQTLs, right? Because remember, we don't have equal numbers of donors, but we have a population of females and males. And you can see the exact same kind of a plot that I showed with quiescent and proliferative cells. These are the females and these are the male donors. Again, for this, for example, gene, you see a very distinct effect on female gene expression, nothing in males. This one is opposite direction, right? In females and males, this G allele is increasing the gene expression and decreasing in males. And again, this one, the same category, there's a difference in the effect size. So just again, like studying, you know, these differences are of course important, because if, not that this will be, but if this were a drug target, right, you might have completely opposite effects, right, in males and females. And again, I told you, and I'm not going to talk about this, but my lab studies sex differences. Women are totally under represented in clinical trials. More cardiovascular drugs have side effects in women than in men. And so this is why I think these are the kind of studies that we need to be studying. So of course, we asked, is there a coronary artery disease locus that shows sex differences? And there was one. So this is a gene called TRF2IP. This is the risk allele. The T allele is the risk allele. You can see that it leads to lower expression of this gene, TRF2IP, in females, and higher expression of this gene in males. And again, we had these phenotypes, right? And one of the phenotypes that also showed sex-dependent effect was calcification, right? T allele leads to lower calcification in females and higher calcification in females. So next we ask, what happens if we actually segregate the data into males and females and look at the coronary artery disease GWAS, right? So here's a locus zoom plot. Again, these SNPs in this locus, when we look at combined both males and females, there's a very strong signal. You can see in the 10 to the minus 15 range for coronary artery disease risk. But when we separate the data into males and females, the signal is coming from males. There's almost no signal in the females, right? There's a locus that displays a very specific sex-biased effect. And this gene is actually turns out to be really interesting because it either acts as a transcription factor or it affects telomere length. I mean, I don't know why a gene would do both, but it does. And if you have lower expression of this gene, you have lower telomere elongation and lower NF-kappa B activation. This is the transcription factor pathway. And remember, we showed lower expression of this gene in females with the risk allele who don't show a coronary artery disease risk, right, in this locus. So it's possible that they actually, you know, are protected because of the low expression of this gene. Okay, so one final small vignette that I'm going to show you is, remember I told you, these loci can affect gene expression or splicing, right? So we asked if these loci, if there are, you know, splicing effects. And basically in this case, we're again looking at the impact of the SNP on splicing, right? So is the allele leading to higher expression of one isoform versus the other? And this is now looking at the entire data set. There are 4,400 genes whose splicing is associated with the genetic variants in smooth muscle cells. This is much lower than the EQTL effect. The EQTLs were 6,500 genes, right? But despite this overall lower discovery, when you look at the coronary artery disease loci, many more, almost twice as many coronary artery disease loci are actually affecting the splicing of the genes, right? And this is a really interesting finding, at least from my perspective. If we're basically saying that the majority of the loci, their molecular mechanism is not gene expression, it is actually splicing of the mRNA. But to be totally honest, I was skeptical of this result because short-read RNA sequencing is notoriously bad at detecting isoforms. So this is, let's say, a gene with 3 different isoforms. You need reads, short reads that span exon-exon junctions to be able to quantify reliably isoforms. And that's hard to do, okay? But I asked, you know, my student who was doing this analysis, if there are variants in known canonical splice acceptor sites, and if this is a real signal, then they should be enriched, right? And actually, that's and this is basically a splice acceptor, splice donor and splice acceptor site. They have a GT or an AG. Like, this is very well established in molecular biology. And basically, he looked at the SNPs and their locations, and what you can see is exactly on top of these GT and AG sites, you see a much bigger, you know, peak, an enrichment of the genetic variants. So this then, at least for some of them, this signal might be real, right? Okay, again, I said short read sequencing, not so good for isoforms. All right, then let's do long read sequencing, right? So we ended up doing long read sequencing where you can sequence cDNA for thousands of bases. It's much more reliable in terms of detecting isoforms. So we took cells from 6 different donors, 3 females, 3 males, and different ancestries, and in different quiescent and proliferative conditions, and basically did long read sequencing. And we discovered the expression of 21,000 genes, but 194,000 isoforms, right? So now I'm starting to believe our results a bit more, you know, maybe like there's some real signal there. So I'll show you just one example. This is a SNP that is associated with coronary artery disease. When you use short read data, this cluster is predicted to be affected by this SNP. And this is our long read data, the one at the bottom. You can see there are many exons in this gene, but there's this one exon exactly where the SNP is that is being skipped in isoform B, right? And basically the risk allele leads to higher expression of one isoform, isoform A, versus the other isoform, isoform B. And this is what you would want to see, right? If there's indeed a splicing effect, you want to see higher expression of one isoform versus the other isoform. Now, the reason I picked this gene is because of Katie, because it's PARP12. I know nothing about PARP12, but PARPs have been studied in macrophages, okay? And if you look at the predictions using AlphaFold, what you find is that this skipped exon is in the catalytic domain of this enzyme, right? So in isoform B, you don't have this catalytic domain, which would certainly be a very important thing about this gene, right? And I don't know if this is real. We're obviously working on this. We're gonna do, antibody staining and this and that. But it seems like the long read sequencing is really helping us to maybe figure out some of the interesting, phenomena that we're observing with the coronary artery disease loci. Okay, so let me just conclude to say that, these loci are really complicated. There are many different molecular mechanisms and different conditions that might be, affecting the disease process. I think we have like many years of work to figure out what each of these loci are really doing, right? There's one person that I really want to acknowledge. So that's my postdoc, Redwan Aherahru, who is finally happy at the end of his experiments. These are all the cell cultures that he did for this work. He's actually now working with Minna Kaikkonen. He has his own group at the University of Eastern Finland. He really tirelessly worked to work on this project. And of course, I would like to acknowledge all of our collaborators. Many people, gave us data and funding sources. And certainly many other folks in the lab helped. It's not just Redwan, and they're all at the top. And many undergrads had a really huge impact on this work. They did aspects of the work. So I want to acknowledge their work as well. And thank you for your time. And I think I'm on time.
No, it's okay. I can get it to you. There you go.
So thank you very much for a really exciting talk. Very excited about your, I can't remember what it's called, TFERP2 IP or RAP1.
Yeah.
And it's very interesting because RAP1 is part of the TNF alpha signaling cascade. Yes. So clearly inflammation is part of this, this mechanism. Do you know if RAP1 interacts with the interferon regulatory factors?
I don't really, honestly.
So because we just reported in Frontiers that a genetic variant near IRF2-BP2, which is a co-repressor of IRF2, is associated with coronary calcification, specifically in men. And so I'm very excited to see your result because you're seeing a sex difference in an inflammatory variant that's also a transcription regulator. So
I will definitely look that up. I mean, the telomere effect also puzzles us. But in vascular cells, it's been shown to affect NF-kappa B signaling. So
it could be it could wear two hats, of course.
It's really interested in this.
Sometimes it's not always the function that it's first identified as regulating, that it's the main factor. That it could be an inflammatory component as well.
Exactly.
So very exciting.
Exactly. Thank you. I think there's a.
Sorry, thank you.
Amazing
talk, thank you.
Thank you.
I just have one question. So how do the quiescent cells in cell culture resemble those healthy contractile cells in vivo?
Yeah. I don't know. That's the answer, honestly. So what we did is, and I might have a slide for this at some point, but we clustered the cells, our gene expression, with the gene expression from the 49 tissues in this GTEx cohort, which is about 900 people, 49 different tissues. And smooth muscle cells, our cells, cluster separately. One would expect that because there's obviously technical issues, but they cluster near aortic tissue and near fibroblastic tissues, etc. So we are presumably capturing some aspect of both the proliferative and the quiescent cells, but I can't tell you that they resemble exactly what's happening in the vessel wall.
Yeah. Thank you. So in the quiescent cell culture condition, you just removed the
FBS,
serum.
Yeah.
Will that affect the cell survival, autophagy, something?
We do it for only 48 hours, and smooth muscle cells can be in a serum-free culture for up to 5 days. And when we obviously looked at differential gene expression between the two conditions, and all the things that you would expect, like calponin, alpha actin, et cetera, they're down-regulated in the things that people have described to be quiescent smooth muscle cell markers are upregulated, if you will, and the proliferative markers are downregulated but we don't see a signal for apoptosis. The main signal we see is proliferation, cell cycle, DNA synthesis, that kind of things.
Great, thank you.
Yeah.
So there's also a question online.
Yeah.
I don't know, it's like hard to read here, but regarding similar genetic studies on cardiac tissues that are affecting rhythm aspects of cardiac functioning. Not sure what you know about that.
There are definitely both heart EQTL studies, and there are cardiomyocyte EQTL studies. There are even IPS-derived cardiomyocyte studies from Kelly Fraser at UCSD and some other groups. I don't think there are any that looked at function, but there are, especially with imaging now, so many GWAS on various aspects of cardiac function. So one could take those loci and overlap, co-localize with the EQTLs from these various heart tissues. There's like left ventricle EQTLs, right ventricle EQTLs, and things like that. Yeah.
So related to that, actually, just get another question here. So when you described the isoforms, and there was almost 200,000 different isoforms, what was the average number of isoforms per gene? And did you look at, does that, I mean, you did some of the functional ones along the way, but then do you look back and say, during calcification, it's primarily not an isoform based change? I mean, proliferation was a big one with the different isoforms.
Yeah.
Did you look at the number per gene or?
We did, and honestly, I don't remember. But I mean, so there are hundreds of thousands of isoforms described in the literature, right? This allows us to actually narrow it down to 200,000 that are expressed in smooth muscle cells. I remember a distribution plot from one of my undergrads who did this study. There are certain genes with 20 isoforms, and there are many genes with one isoform. I can't remember the average, honestly.
It'd be interesting to see if that, if those genes are the stronger ones proliferatively, or if those are, anyway.
Right. One quick thing so when we're looking at the isoform effects, we're actually looking at the loci that don't display an EQTL effect. Because we don't want to confuse the two things, right? We are only, so there are 164 SQTLs and 85 EQTLs. So we're looking at the subset that is not an EQTL, because it's hard to dissect then if it is an overall gene expression effect, isoform expression effect, et cetera, et cetera.
Okay, Peter.
Well, this is fantastic talk. And so two things I was wondering. One is that, the one you were very much, elucidating, I said, SNHG 18, I think it's being associated with microvascular disease in diabetes, in some of these conditions. And I'm trying to figure out, do you also down the road look at, I guess, neighborhood cell types, because ultimately compare, data sets, from smooth muscle cells, with whether it's endothelial or, sort of cardiac myocytes. And then the other question is not related to that, is that we look a lot at the protein expression as well. Does protein expression help, in terms of ultimately, sort of assigning some of the potential functional, sort of significance?
Okay, 2 quick answers. So all those SNH18 studies came out after we started. So look at their publication dates. They're all after or around the time we put this paper on bioRxiv. Okay, basically, if you look at single cell data in at least coronary arteries, it's very smooth muscle cell specific. We have not looked at cardiomyocytes, etc., other things. For your second question, we looked at secreted proteins, not the proteins in the cells, but those that were secreted from the cells, because smooth muscle cells secrete cells, extracellular matrix, etc. And there are some loci that affect the abundance of secreted proteins. Not as many. Like we're actually now working on a paper on LTBP1. It is in the TGF beta pathway, and it's a secreted protein. It's one of the loci it's affecting that. But having said that, those of you who have done these kind of things, it's well established in the field that whether you look at C. elegans or yeast or mice, if you look at transcripts and then proteins and then metabolites, as you go up the layer, the genetic effect actually gets buffered out. So you see more genetic impact on transcript abundance and less on protein and less on metabolites. And so while especially serum proteomics is extremely important to identify these loci, you find fewer and fewer co-localized loci with proteomics, basically. And that's a very well-established phenomenon, like even in Arabidopsis and all kinds of things.
Interesting. Okay, any other
Wonderful questions?
Oh,
yeah, go ahead.
It was very impressive talk. I really enjoyed it. So I have 2 questions. First is the older smooth muscle cell EQTL or SQTL you found. Is it very specific for smooth muscle cell? And then also the sex difference you found is a cell specific smooth muscle cell specific, or actually happen in other possibly organs or cell type? Second question is maybe you're doing it, whether you had a chance to look at how SQTL affect the chromosome structure possibly in interactions.
Yeah, no, we have not looked at that. But I have a slide to answer your first question. And I'll think I might be able to show it to you in 1 second. And this is why I would, okay. So we looked at GTEx, right, which is basically the largest EQTL catalog. Like I said, 900 people, 45, 50 different tissues. So these are the overlap of our smooth muscle cells, and these are the ones that we're identifying, right? So these are only in our data sets, basically. And there are definitely smooth muscle cell specific EQTLs that overlap CAD loci. You find those signals obviously in the aortic tissue as well, right? Yeah. Oh God, did I forget to say, sex specific. We have not looked at other tissues actually. Yeah, so I can't really say much about that. Go ahead.
These are sex-specific differences of your cultured cells.
Yes.
And everyone should know that cultured cells retain sex-specific differences in culture, whether you culture them or more or not, right?
Oh, for sure. I mean, you have the presence and absence of the Y chromosome, right? At least, yeah.
So
then there's the X inactivated or X active, like region, which
I was going to actually ask that about your expression analysis versus your genetic analysis, because your genes do not include the Y and X sequences, right? Did you find anything just at your transcript level that's specific to X and Y that you might be able to go back? Because I know, as far as I recall, most genetic studies don't include the sex chromosomes at all. And so when you have anything expressed from there, these EQTL studies sort of fall off the wagon a bit.
They do, yeah. We excluded the XX chromosome SNPs too, for mostly analytical reasons, and it's hard to impute them. But actually now there are methods to do it.
Okay.
And now that I have one grad student who's actually really interested in sex differences, we're definitely going to do those too.
Yeah, that's very cool.
But interestingly, if you look at CAD GWAS, and when you do a sex-biased analysis, there are no variants on X or Y chromosomes that are associated. Yeah, it's all in the autosomal chromosomes.
Interesting. Okay.
Yeah.
Well, thank you so
Thank
much.
you
Mete. What an
all.
excellent
Thank you.
talk. Thank you, everybody, for joining. Thank you online.
And thank you, everyone who was online.
Yes.